Hello everyone, and welcome to today's presentation. I'm excited to have you here as we explore the fascinating world of carbohydrates metabolism. The title of our talk is BCHM 201 Carbohydrates Metabolism. Over the next few minutes, we'll cover the key aspects of how carbohydrates are processed in the body, including their pathways and significance. So, let's dive in and get started. All living cells need energy to perform their essential activities. This energy isn't just floating around. It's stored in the chemical bonds of organic molecules, like carbohydrates, fats, and proteins, which we get from the food we eat. When we consume food, these molecules are broken down through enzymatic reactions inside the cells. This process releases energy, which is then captured in the form of ATP or adenosine triphosphate. ATP is essentially the cell's energy currency. It powers all the fundamental processes that keep the cell alive and functioning. So, understanding how cells generate and use this energy is key to grasping how life operates at a cellular level. The food we eat is mainly made up of three key components: proteins, polysaccharides, which are basically carbohydrates, and fats. When we consume these nutrients, our body first breaks them down into smaller, more manageable units. Proteins are broken down into amino acids, polysaccharides into sugars, and fats into fatty acids and glycerol. This initial digestion process happens outside the cells, in our digestive tract. Once these smaller molecules are formed, they enter our cells. Inside, they undergo further processing through oxidation. For amino acids, sugars, and fatty acids, this happens via different pathways: glycolysis in the cytosol and the citric acid cycle inside the mitochondria. These processes are crucial because they generate ATP, our body's main energy currency, from an inorganic phosphate. This entire sequence, breaking down food, absorbing nutrients, and generating energy, is fundamental to how our bodies function and connect to the overall theme of energy metabolism. In glycolysis, a molecule of glucose is broken down through a series of enzyme-driven reactions. These reactions convert glucose into two molecules of pyruvate, which is a 3-carbon compound. As these reactions occur, some of the energy released from glucose isn't lost, but is instead captured in the form of ATP and NADH, which are crucial energy carriers in the cell. Glycolysis is really fundamental because it's almost a universal pathway across different organisms. It serves as the main route for glucose breakdown, handling the largest flow of carbon in most cells. That's why it's often called a central pathway in metabolism. In some tissues and cell types, glycolysis is actually the only way they generate energy. For example, in red blood cells, the kidney medulla, the brain, and sperm cells, glycolysis provides all the metabolic energy they need. This pathway, known as the Mdenmayer-Hoff pathway, is essential for cellular energy production, connecting the breakdown of glucose to the generation of energy that powers cell functions. Now, let's look at some interesting examples of how plant tissues are modified for energy storage. For instance, potato tubers are a great example. They store starch, which is a form of energy reserve. Similarly, some aquatic plants like watercress derive most of their energy through glycolysis, especially in low-oxygen environments. It's also important to note that many anaerobic microorganisms, those that thrive without oxygen, are completely dependent on glycolysis for their energy needs. Glycolysis is really fundamental in these contexts because it allows these organisms to generate ATP without oxygen. This brings us to fermentation, which is a broad term that describes the anaerobic breakdown of glucose or other organic nutrients. Essentially, fermentation enables organisms to produce energy, ATP, when oxygen isn't available. So, in summary, glycolysis and fermentation are crucial pathways for energy production, especially in environments where oxygen is scarce or absent. They connect directly to our overall theme of how cells adapt their energy metabolism to different conditions. Let's walk through the key steps of glycolysis, starting with the first one. Glucose is phosphorylated to glucose 6-phosphate, and the enzyme responsible for this is hexokinase. This enzyme uses ATP, splitting it into AD, and attaching the pie to the glucose. The energy released from ATP hydrolysis actually drives this reaction forward. Hexokinase is a crucial enzyme in glycolysis, and importantly, this step is irreversible, meaning it commits the glucose to the pathway. Next, glucose 6-phosphate is isomerized into fructose 6-phosphate by phosphohexose isomerase. This rearrangement prepares the molecule for the subsequent phosphorylation step. Then, fructose 6-phosphate is further phosphorylated to fructose 1,6-bisphosphate. The enzyme here is phosphofructokinase, which is another key regulatory enzyme. Just like the first step, this one is also irreversible, emphasizing its importance in controlling the pathway. Moving on, fructose 1,6-bisphosphate is split into two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, by aldolase. These two molecules are interconvertible. Dihydroxyacetone phosphate is isomerized into glyceraldehyde 3-phosphate by phosphatriose isomerase. This split sets the stage for energy extraction in the subsequent steps. Finally, glyceraldehyde 3-phosphate undergoes dehydrogenation and phosphorylation, transforming into 1,3-bisphosphoglycerate. This process involves NAD+ as a coenzyme and is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. This step is crucial because it captures high-energy electrons and prepares the molecule for ATP generation later in glycolysis. Now let's look at what happens next in glycolysis. First, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. This reaction is catalyzed by the enzyme 1,3-bisphosphoglycerate kinase. An important point here is that this process actually produces ATP directly through what's called substrate-level phosphorylation. So essentially, we're generating energy right at this step. Moving forward, the 3-phosphoglycerate undergoes an isomerization to become 2-phosphoglycerate. This involves shifting the phosphate group from the third carbon to the second, and the enzyme responsible for this is phosphoglucomutase. Next, 2-phosphoglycerate is converted into phosphoenolpyruvate, or PEP, by the enzyme enolase. During this step, a water molecule is removed, which results in the formation of a high-energy phosphate bond in PEP. It's also worth noting that enolase requires magnesium ions, Mg++, to function properly, and its activity can be inhibited by fluoride ions. Then, phosphoenolpyruvate is dephosphorylated to pyruvate by the enzyme pyruvate kinase. This step is critical because it generates another ATP molecule. Importantly, this reaction is irreversible, making it a key control point in glycolysis. Finally, under anaerobic conditions, pyruvate is reduced to lactate by lactate dehydrogenase. This allows glycolysis to continue when oxygen is scarce. Conversely, under aerobic conditions, pyruvate enters the citric acid cycle for complete oxidation, producing much more energy. The lactate produced during anaerobic glycolysis can then enter the Cori cycle, where it's converted back to glucose in the liver. Understanding these steps highlights how glycolysis efficiently produces energy and adapts to different cellular conditions. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about how the flux of glucose through glycolysis is tightly regulated to keep our ATP levels pretty much constant. This regulation is crucial because ATP is our cell's main energy currency, and maintaining its balance is vital for cell function. At the same time, glycolysis also supplies intermediates necessary for biosynthesis, so it's a finely tuned process. The adjustment in glycolytic rate happens through a complex interplay involving ATP consumption, which directly impacts how fast glycolysis proceeds, and NADH regeneration, which is essential for maintaining the pathway's flow. Additionally, several key enzymes, like hexokinase, PFK1, and pyruvate kinase, are regulated allosterically, meaning their activity can be quickly turned up or down based on the cell's needs. This regulation isn't just about enzyme activity. It also involves fluctuations in metabolite concentrations that reflect the cellular balance between ATP production and consumption. Think of these as real-time indicators that help the cell adjust glycolysis on a second-to-second basis. On a longer time scale, hormones play a big role in regulating glycolysis. Hormones like glucagon, epinephrine, and insulin influence the process by affecting gene expression for various glycolytic enzymes. This way, the cell can adapt its metabolism to different physiological states, like fasting or after a meal. Overall, glycolytic regulation is a perfect example of how our cells dynamically respond to their energy demands and metabolic needs, ensuring everything runs smoothly and efficiently. Under aerobic conditions, the end product of glycolysis is pyruvic acid. The next step is the formation of acetyl coenzyme A, or acetyl-CoA now. This step isn't technically part of the citric acid cycle itself, but you'll see it shown on diagrams to connect the processes. Acetyl-CoA, whether it comes from glycolysis or from the breakdown of fatty acids, is really the starting point for the citric acid cycle. Think of it as the bridge that links carbohydrate metabolism to the cycle. The cycle begins when a 4-carbon compound called oxaloacetic acid reacts with acetyl-CoA, which has 2 carbons. Together, they form citric acid, which has 6 carbons. The main goal of each turn of the cycle is to produce 2 molecules of carbon dioxide. This process involves a series of oxidation reactions, where hydrogen and electrons are lost at 4 specific points. These electrons don't just disappear. They're transferred to the electron transport chain, which is where a large portion of ATP is generated. So, to sum it up, the citric acid cycle is a crucial part of cellular respiration. It not only helps us extract energy from nutrients, but also prepares that energy for the production of ATP, which powers nearly all cellular activities. Let's walk through the first step. The acetic acid part of acetyl-CoA combines with oxaloacetate to form citrate. Think of acetyl-CoA as a shuttle. Its main role here is to transport acetic acid from one enzyme to another. Once this transfer is complete, the CoA molecule is released through hydrolysis, freeing it up to pick up another acetic acid and restart the cycle. This process is crucial because it keeps the cycle moving smoothly. Moving on to step 2, the citrate molecule undergoes a transformation called isomerization. Essentially, a hydroxyl group and a hydrogen are removed from citrate, forming water. During this process, the 2 carbons involved form a double bond. Then, water is added back, but in a different position, which effectively reverses the original arrangement. This results in the formation of isocitrate. This step is important because it prepares the molecule for the next oxidation phase. Now, in step 3, the isocitrate is oxidized by NAD. Here, NAD acts like a carrier, accepting a hydrogen atom and a hydroxyl group from isocitrate. When NAD accepts the hydrogen, it becomes reduced, turning into NADH. The removal of the hydroxyl group creates an unstable intermediate, which then releases a molecule of carbon dioxide. This results in the formation of alpha-ketoglutarate. This oxidation step is vital because it helps release energy and prepares the molecule for further processing in the cycle. Now, moving on to step 4. Here, coenzyme returns to the scene to help oxidize the alpha-ketoglutarate molecule. During this process, NAD is reduced again to form NADH, and it leaves with another hydrogen atom. This movement creates some instability, causing a carbonyl group to be released as carbon dioxide. In its place, a thioester bond forms between what used to be alpha-ketoglutarate and coenzyme A, resulting in a molecule called succinyl coenzyme A. Next, in step 5, a water molecule comes in and sheds its hydrogen atoms to coenzyme A then. A free phosphate group displaces coenzyme A and bonds with the succinyl complex. This transfer of phosphate to GDP produces GTP, which is an energy-carrying molecule, and the process leaves behind a molecule called succinate. In step 6, succinate undergoes oxidation by FAD, or flavin adenine dinucleotide. The FAD removes 2 hydrogen atoms from succinate, which causes a double bond to form between 2 carbon atoms, creating fumarate. This step is crucial because it helps transfer electrons for energy production. Moving into step 7, an enzyme adds water to fumarate, converting it into malate. This involves adding a hydrogen atom to one carbon and a hydroxyl group to an adjacent carbon, transforming the molecule into malate. Finally, in step 8, malate is oxidized by NAD, turning the hydroxyl group into a carbonyl group. The end product of this cycle is oxaloacetate, which can then combine with acetyl-CoA to restart the Krebs cycle again. This sequence of steps highlights how energy is extracted and stored efficiently during cellular respiration, keeping our cells fueled and functioning smoothly. This slide doesn't contain enough information. That's why we're moving to the next slide. The goal of glycolysis, glycogenolysis, and the citric acid cycle is to conserve energy as ATP from the breakdown of carbohydrates. When the cells have enough ATP, these pathways are actually turned down or inhibited. It's like the cell recognizes it has enough energy and doesn't need to produce more. In those situations of excess ATP, the liver shifts gears and starts converting other molecules into glucose or glycogen for storage. This is where biosynthesis of glycogen, or glycogenesis, comes into play. It's essentially the process of building up glycogen stores when energy is abundant. This process ensures that the liver can store excess glucose efficiently, maintaining energy balance and preparing for times when energy might be scarce. So, in essence, when energy is plentiful, the body switches from breaking down molecules to storing them, highlighting the dynamic regulation of metabolism based on the cell's needs. Glycogenesis is the process where we make glycogen from glucose, and this process really depends on the body's needs for glucose and ATP, which is our energy currency. When both glucose and ATP levels are relatively high, the hormone insulin kicks in and encourages the body to store excess glucose as glycogen, mainly in liver and muscle cells. This storage mechanism is crucial for maintaining energy balance. Now, when it comes to building glycogen, each glucose molecule that gets incorporated into the glycogen chain requires one ATP molecule. This energy investment supports the formation of the complex, branched structure of glycogen. An interesting point here is that glucose 6-phosphate acts as a kind of crossroads compound. It's a key intermediate in this process. It can be synthesized directly from glucose or produced at the end of gluconeogenesis, which is our body's way of creating glucose from non-carbohydrate sources. So, in summary, glycogenesis is tightly regulated based on energy status and hormonal signals, ensuring that excess glucose is safely stored for future needs. This process links directly to our overall theme of how our body manages glucose and energy efficiently. This slide doesn't contain enough information. That's why we're moving to the next slide. In glycogenolysis, the process by which our body breaks down glycogen, the stored form of glucose, primarily in the liver and muscles, is quite fascinating. First, glycogen is converted into glucose 1-phosphate, and then this molecule is transformed into glucose 6-phosphate. This step is crucial because glucose 6-phosphate can then enter various metabolic pathways to meet the body's energy needs. Two key hormones regulate this process: glucagon and epinephrine. Glucagon is a peptide hormone released from the pancreas, especially when blood glucose levels are low, signaling the body to release stored energy. Epinephrine, on the other hand, comes from the adrenal glands and is released in response to stress or danger. Think of it as the body's fight-or-flight hormone. Both hormones work by activating specific enzymes. They stimulate glycogen phosphorylase, the enzyme responsible for breaking down glycogen, to kick off glycogenolysis. At the same time, they inhibit glycogen synthase, which is the enzyme that promotes glycogen synthesis, effectively stopping glycogenesis when energy is needed. Now, glycogen itself is a highly branched polymeric structure, with glucose molecules as its basic units. When the body needs energy, the process begins by hydrolyzing individual glucose units from the glycogen chain. This initial step involves removing glucose molecules and attaching a phosphate group at the C1 position, creating glucose 1-phosphate. In the next step, this phosphate group is shifted from the C1 to the C6 position, converting glucose 1-phosphate into glucose 6-phosphate. This molecule acts as a crossroads, either entering glycolysis to produce energy or, in the liver, being converted into free glucose to maintain blood sugar levels. So, to sum up, glycogenolysis is a finely tuned process, controlled by hormones in response to our body's needs, ensuring we have energy when we need it most. Now, let's take a closer look at what happens when glycogen is our carbohydrate source and energy is needed. The first step in glycolysis in this case is the formation of glucose 6-phosphate. This molecule is crucial because it essentially acts as the gateway into the glycolytic pathway, where glucose is broken down to produce energy. However, if the body doesn't need energy immediately, glucose 6-phosphate doesn't just stay put. Instead, it can be converted back into glucose. This free glucose then enters the bloodstream and is distributed to various cells throughout the body, including the brain. This process highlights the body's ability to balance energy production and storage, ensuring that vital organs like the brain have a steady supply of glucose when needed. It's a great example of how metabolic pathways are interconnected, maintaining homeostasis and supporting overall function. This slide doesn't contain enough information. That's why we're moving to the next slide. Gluconeogenesis is a fascinating metabolic pathway that essentially allows the body to produce glucose from non-carbohydrate sources. These sources include pyruvate, lactate, glycerol, and certain amino acids known as glucogenic amino acids. It's a crucial process, especially when carbohydrate intake is low or during periods of fasting. Most of this activity occurs in the liver, which is the body's main metabolic hub. But the kidneys, specifically their cortex, also play a significant role, just to a lesser extent. This process becomes particularly important during fasting, starvation, or intense exercise when glucose levels need to be maintained for energy. It's important to note that gluconeogenesis is highly endergonic, meaning it requires energy input to proceed. And you'll often see it linked to ketosis, because during these energy-deprived states, the body shifts to producing ketone bodies as an alternative fuel source. In essence, gluconeogenesis is part of the body's remarkable ability to maintain blood glucose levels and energy homeostasis under various physiological conditions, tying into the broader theme of metabolic regulation and adaptation. Fatty acids in most animals cannot be converted into glucose. An interesting exception to this is odd-chain fatty acids, which can produce propionyl-CoA. This compound is a precursor for succinyl-CoA, which can enter the citric acid cycle and eventually contribute to glucose synthesis. Now, in plants, especially during seedling development, there's a different pathway at play. The glycosylate cycle allows plants to convert fatty acids, specifically acetate, into a primary carbon source for growth. This cycle produces 4-carbon dicarboxylic acids, which can then feed into gluconeogenesis, helping the plant generate glucose from stored fats. Glycerol, which is part of all triacylglycerols, can also be used for gluconeogenesis. However, it's important to note that in organisms like humans and other mammals, glycerol that originates from glucose isn't typically considered a true gluconeogenic substrate. That's because, in these cases, glycerol can't be used to generate new glucose molecules directly. So the key takeaway here is that while animals have limited ability to convert fatty acids into glucose, plants have a specialized pathway, the glycosylate cycle, that enables them to do so. And glycerol can contribute to gluconeogenesis, but its role varies depending on the organism and its metabolic context. This highlights the diversity in how different organisms handle fats and sugars, tying back to our overall theme of metabolic flexibility. Let's explore how gluconeogenesis kicks off inside the mitochondria. It all starts with pyruvate, which undergoes a process called carboxylation to form oxaloacetate. This step requires energy, specifically one molecule of ATP. The enzyme responsible is pyruvate carboxylase, which is actually stimulated when there's high levels of acetyl-CoA, typically during active fatty acid oxidation in the liver. Conversely, if ADP levels are high, this reaction is inhibited, helping regulate the process. Now, oxaloacetate can't leave the mitochondria as it is. So, it gets reduced to malate using NADH, which allows it to be transported out of the mitochondria efficiently. Once in the cytoplasm, malate is oxidized back to oxaloacetate using NAD+, setting the stage for the next steps. The oxaloacetate then undergoes decarboxylation and phosphorylation, transforming into phosphoenolpyruvate, or PEP, thanks to the enzyme phosphoenolpyruvate carboxygenase. This reaction also consumes GTP, converting it into GDP. These steps are crucial because they link the mitochondrial and cytoplasmic phases of gluconeogenesis, enabling the liver to produce glucose when needed, especially during fasting or intense exercise. Now, moving on to the next steps in gluconeogenesis, it's interesting to see how they mirror the reactions of reversed glycolysis. But there's an important enzyme here, fructose 1,6-bisphosphatase, that plays a crucial role. It converts fructose 1,6-bisphosphate back into fructose 6-phosphate. This step is particularly important because it helps overcome a large negative delta, or free energy change, which would otherwise make the reaction unfavorable. From there, the process continues with phosphoglucose isomerase, converting fructose 6-phosphate into glucose 6-phosphate. This molecule is quite versatile. It's a key intermediate that can either go towards generating free glucose or be used in other metabolic pathways, depending on what the cell needs at the moment. Now, it's important to understand that free glucose isn't automatically produced during gluconeogenesis. Unlike glucose 6-phosphate, free glucose tends to diffuse out of the cell easily, so the body needs a specific process to produce and export glucose efficiently. That brings us to the final step, which is quite unique. It occurs in the lumen of the endoplasmic reticulum. Here, glucose 6-phosphatase hydrolyzes glucose 6-phosphate to release free glucose. This glucose is then transported into the cytosol by specialized glucose transporters embedded in the ER membrane, making it available for use elsewhere in the body, like in the bloodstream. So, in summary, these steps ensure that glucose is produced efficiently and can be exported from the liver cells to maintain blood sugar levels, which ties back to the overall theme of energy regulation and homeostasis. This slide doesn't contain enough information. That's why we're moving to the next slide. Now, while most steps in gluconeogenesis are actually the reverse of those in glycolysis, there are 3 key reactions that are different, mainly because they are highly regulated and strongly exergonic. These reactions are replaced with alternative, more kinetically favorable ones to make the process work efficiently. Specifically, the enzymes involved in glycolysis, hexokinase or glucogenase, phosphofructokinase, and pyruvate kinase, are replaced by glucose 6-phosphatase, fructose 1,6-bisphosphatase, and PEP carboxygenase in gluconeogenesis. This setup creates a reciprocal control system, which is crucial because it allows glycolysis and gluconeogenesis to inhibit each other, preventing what we call a futile cycle, where both pathways would waste energy by running simultaneously without productive output. Ultimately, the rate of gluconeogenesis is controlled by a key enzyme, fructose 1,6-bisphosphatase. Its activity is finely tuned through signal transduction pathways involving CAMP and phosphorylation, allowing the cell to respond dynamically to different metabolic needs. So, this regulation ensures that the body efficiently manages glucose production and utilization, aligning with the overall theme of metabolic control and energy balance. Lactose intolerance is a common digestive issue that many people experience. It happens when the body can't properly digest lactose, which is a sugar found mainly in milk and dairy products. To break down lactose, our bodies normally use an enzyme called lactase. This enzyme splits lactose into 2 simpler sugars, glucose and galactose, that can be easily absorbed into the bloodstream. Think of enzymes as proteins that help speed up chemical reactions in our body. Now, in cases of lactose intolerance, the body doesn't produce enough lactase. So, lactose remains in the digestive system instead of being fully broken down. This undigested lactose then ferments with bacteria in the gut, kind of like how yeast ferments to make beer. This fermentation process is what causes the uncomfortable symptoms associated with lactose intolerance, like bloating, gas, and cramps. It's also worth noting that lactase levels tend to decrease as we get older, and certain health conditions can further reduce its production. So, lactose intolerance is often something that develops over time and can vary from person to person. Understanding this helps us see how enzyme activity directly impacts digestion and overall health, which ties into our broader discussion of how our bodies process different nutrients. In the glycolysis pathway, glucose is aerobically converted to carbon dioxide and water, and anaerobically converted to lactate. All the reaction steps take place in the cytoplasm of the cell. Glycolysis is the only source of energy in anaerobic cells, where oxygen is not available. The number of ATP molecules gained per glucose molecule under aerobic conditions is about 36, while under anaerobic conditions, it's only 2 ATPs. As for the Krebs cycle, it yields approximately 2 ATPs per mole of glucose. These points highlight how energy production varies depending on oxygen availability, which is a key aspect of cellular metabolism and overall energy management in the body.